So Christian gave, I think, a great overview of just kind of what the initial kind of using Speckle at its typically its most basic form, and from an engineering perspective, what you can start doing with it in terms of integrating it into different devices for different ends. And that's, I think, a theme of a lot of, I think it's a lot of instrumentation in general, but also with Speckle in particular is you can work with it very simply if your application is that's all it requires. And if you need some more information out of it, it really is this concept of unpeeling the layers of the onion. That's how we're going to focus on this today. There are a number of equations here, but I'm going to try to put them into just the general context. I'm not going to pretend that I know how all of them are derived or anything like that. It's more of a this is kind of where the field has taken this sort of quantitation, and so it's an awareness more than anything else. So really the take home message is going to be kind of this concept of we have these reflectance images. So we can do with one image, which is one of the beautiful things with speckles. You can just take a 10 millisecond image and that's all you need. You can take a sequence of images. And so we're going to kind of go back and forth with those concepts. Typically, if you have the luxury of a little bit of time, you take a sequence of images, you process each one as you would process a single image, and then just average them to reduce some of the noise. So that's typically what we do, but you can do it with one image. Then somehow we get contrast. And so Christian talked about the sliding window, and that's the technique we typically use. There are other ways to do it, and I'll talk about one other way in particular, or actually a couple other ways in particular. But somehow you go from the raw speckle image mode to the speckle contrast world. And then you need some way, this is where a lot of the tricks, a lot of the theory comes into play, is you need some model-based way of taking a speckle contrast image and turning it into something that represents flow or particle motion of some sort. And that's where the theory can get pretty hairy pretty quickly because we are dealing with an interference pattern. And so we're going to be talking about two parameters in particular, G1 and G2. They're related to each other. They're going to be used a little bit, not interchangeably, but you're going to see some cases where there's going to be G1, some where there's going to be G2. Basically, G1 is electric field dynamics, G2 is the intensity dynamics of the electric field squared. And you're going to, and there's a very simple equation that links the two together. So if you can understand it for G1, you can go to G2. If you can kind of grasp it for G2, then you can go to G1. Okay. But it's all speckle and how the speckle pattern changes and this concept of correlation of the speckle. And then we get by applying whatever model you want to do, however simple you want it to be, however complicated you want it to be, you get some map, ideally some map of flow. And it can be semi-quantitative, like what Christian was talking about, was a lot of it was it's quantitative, it gives you a number, but it's not an absolute number. Or what he talked about with this diffusion coefficient, trying to correct the flow for optical properties, you can get to a value that's we'll call it quantitative. And there's really trade-offs between simplicity and robustness. For a lot of things, simple works great. And I think I took out the Goodman slide. So a lot of this is from like the 1960s. Joseph Goodman, who is a professor at Stanford, I think he's retired now, but he pioneered a lot of this concept of speckle is it's not noise. You can actually infer information. He was looking at radar to start with, and then he moved to optics. 45 years ago, Adolf Erker and David Bryers were the pioneers of this. They pioneered the concept of taking those concepts and putting them into biological tissues. They started in the retina, and then Dr. Bryers eventually moved to skin, and just to show some demonstrations of it. Technology limited, right? They didn't have CCD cameras in early 1980 that were something that you could just buy relatively cheaply. So actually, the first example in this paper is they did film photography, and they basically put optical filter, like spatial filters in to try to give you a map of retinal blood flow. So yeah, there was no image processing in this first paper. It's just all theory and then a measurement and then an image. So the idea here is that you take a picture in this paper, take the picture, And then if you understand the spatial statistics and how it relates to whatever the speckle pattern looks like, you can then infer an image of flow. Okay. So concepts from, from 40, more than 40 years ago. And so this equation, it's been corrected since then, but I think it's a nice equation to start with. So what's going on in this equation? You see an integral. One of the things that's nice and easy is the T is the exposure time of your camera. So that's your shutter. So that's pretty straightforward. You can dial that into a camera. But now you got these other parameters in here as well. And what it's basically saying is that if I take an image or a local region of pixels in the camera, and I look at the speckle pattern, I look at the speckle pattern, I look at the variance, or Christian was talking about standard deviation. Obviously, variance is standard deviation squared. So if you look at just how the pattern varies in a small area, it's kind of a remarkable thing. Based off of that, you could then infer how that speckle pattern is changing over time at that local area. So you get an image, and you just from a single image and the statistics of that region of that image, you can actually infer time-wise what's going on with some limits. But it's kind of a bizarre it's a bizarre concept. But that's the same thing like with the image that showing the train moving. You could look at a small area of that image, and you could pretty much, if I showed you two areas of that image, like one on the person and one on the train, and I said which was moving faster or which was moving, you probably can guess it with, you probably would give me the right answer. And so qualitatively, there is this inferment you can get just by looking at a small area of an image. And in our case, we're talking about the visibility of the speckles. Okay. So going back to here, so we have the time variance over some exposure time. We integrate over that, and we can get the local speckle contrast or speckle variance, and vice versa. And so, this is one of the this is a paper from the 90s by David Breyers, and simple relationship. It's the speckle contrast that Christian showed. You take standard deviation over a small area divided by the average intensity over that small area, and you can get this contrast value. Okay, and so this right here is basically related to this right here. Okay. Yeah, article. This is an integration. This is basically an integration term here. So basically, at specific times, what is it? So we'll get into this term in a second. But this is related to the intensity measurement. Okay. So going into a little bit of the theory. So this is just showing two waves here are with the same frequency, slight offset. So we have amplitudes and we have these phase differences right here. And so with speckle theory, the assumption is made that if you have a bunch of waves hitting like a single point, and those waves have a uniform phase distribution, so there's an equal chance for me to get this difference as there is to get this difference, as there is to get no difference, etc. Okay. So if you have this condition met, then one of the things you're going to infer from it is the speckle pattern is a truly random pattern. So speckle is a statistical optics technology. So, or it's a technology based in statistical optics. So that's why the comment that Bruce Stromberg made about this is the most complicated technology. It's statistical, in reality. And so, at least at the level that we can infer things, it's deterministic. If you go down to the level, like basically maybe at the nanometer level, and you can freeze things in time, you can maybe get a deterministic piece of information from it. But from our purposes, things are happening in such a we can't measure at that level. It's called a fully evolved speckle pattern if this condition is satisfied. And I'll show you what that can look like. And the interesting thing is that this means that if I look at all the intensities in the image and I just plot it as a probability distribution, it follows a negative exponential. And this is what's called a Lorentzian distribution. So this is all theoretical. And if I calculate the standard deviation of this entire image and divide by the mean of this entire image, I'm going to get a value of 1. So this is the upper pure limit of speckle contrast. It's 1. So this is for a perfect speckle pattern. So here's an example of a pattern. This is actually generated computationally. You guys will be generating patterns in the lab. And this is a semi-log graph, so that's why it looks linear when at first glance. But this is an exponential decay, and basically this is just showing you take the intensity values of all the over here and just plot them. You divide by the maximum and just plot them over here. You're gonna get something that follows a negative exponential decay. Okay, perfect conditions. In reality, we don't measure under perfect conditions, but this is the theory. So if you correct that original integral equation, and then you solve for contrast. So this is speckle contrast squared. So it's related to the speckle the variance of the pattern. You then get this equation here. So exposure time is still here, which is great. And then we have the C term here, which is I didn't talk about what it was before, but this is the temporal autocovariance. And I'll talk a little bit about what that means in a second. And you just have that this is the average intensity. So the average intensity squared of a region, let's say. So once again, spatial statistics over here, temporal statistics over here. Okay. So what is this temporal autocovariance? Does anybody know? Ezekiel. Yes, you are correct. So it's the measurement of it's the relationship between the intensity kind of at a certain time point or at a time point and then at some delay. Okay, so it would be like if you had let's say a sine wave, and then then you push a button and the sine wave frequency starts changing. Over short time periods, it'd be correlated, and over longer time periods, it would get worse and worse. The correlation between what it was at one point in time versus some time delay later. Okay, so yeah, Anna, yeah.
How the signal is correlated to itself.
Why don't we need one? So
Um,
we got a long coherence time.
yes. I don't need the hearing place. I'm sorry, my brain.
Oh, we do need a coherent light source. Yeah, in order to do this, yeah, you need a coherent source. Yeah. Oh, yeah. This has this we use so there's like, but the question I guess becomes how coherent does it need to be, right? So I think Christian alluded. I mean, people actually Andy Dunn's group showed, and we've seen this too, is that with a laser pointer you can get this to work. Is it great? No. Is it good enough? Maybe. So, but yeah. So there's some of those long coherent sources that go like into diffuse correlation spectroscopy. We use those, but you don't need a laser that good necessarily to do this. Okay, so here's just an example. So we have intensity fluctuating over time. So kind of something kind of simple here. And then this black line here represents the average value. So we have our intensity at some time point. We have our average intensity over here. We have intensity at some time point later, and then subtract off the average intensity. So it's a ratio of the intensity at two different time points with the average intensity subtracted off. So just kind of just going through this pictorially. So, if we have basically looking at this graph here and we start looking at two different time points, you can kind of see that basically we're doing this for some time delay, but we're doing it at all time points, t. Okay, so we're changing t, and then t plus tau shifts at the same distance, and then we could do this by changing the delay. So we can look at this point, and then just kind of move that over. We can look at this point, move that over, etc. So we look at all these delays at all these different time points, and then we do basically an integration of this equation. So that's just numerically what's going on under the hood. Okay, and then if we do that, we can get contrast. Now this requires, the reason why we don't do this in general in practice is because this requires a really fast camera. These sort of dynamics are they're very short, and so we'll get into that part later. But if you want to do this with a your cell phone, or you want to do it with just some standard CCD camera in the lab. You can't, you're not gonna be able to measure this adequately. So this is just kind of just showing here if we have something fast varying, here are something slower varying, in terms of the intensities. So these are just just some random or just some distributions. Then you could get, if you look at the auto covariance, you can get something that looks like this in terms of if you just calculate this right here and just apply it to the intensity signal. So the question I have is, which one here looks like the faster varying intensity? So I'm blocking off, if you kind of can see on the graphs, I'm blocking off, it's symmetrical around zero. So if you just focus at zero and the positive time delays, which one's delaying fast? Which one's decaying? Which one's got faster varying intensity? Which one do you think? The one on the right. Why? So if it's decaying slower, then so basically, if it's varying more quickly, it's going to be less similar to itself more quickly. So like, actually, it's the opposite. It's in this case, this is the faster varying one, but it's the faster decay. So it's less. So when something changes quickly, it's just less similar to itself more quickly. So that's the reason why. But thank you. Um, and what this will end up being is that if this is decay more quickly versus decay more slowly in this case, just the area under the curve is lower. So this will then just mathematically, because it's decaying faster, will lead to a lower contrast. This is decaying more slowly, so it'll lead to higher contrast. Okay, so faster, and I think Christian alluded to this, lower contrast, faster flow, or higher contrast, lower flow. And so that's a theme consistently throughout. Okay. And so this is the covariance and relationship. So G1 was the electric field correlation function. G2 is the intensity correlation function. So if you know the mean intensity and you have the covariance graph, you can calculate G2. So once again, if you can measure fast enough, you can measure G2. And G2 is related to G1 it's this thing called the secret relation. So you'll see this a lot if you look at diffuse correlation spectroscopy and some of the more theoretical speckle papers, you'll see this relationship. Basically, this is a fairly simple relationship between what you measure intensity wise and then what you measure electric field, or the dynamics of the intensity, the field, and the dynamics of the electric field. Okay. And then there's this, this nice multiplicative factor here, beta, which is basically just it's a lump sum term of, it's a fudge factor basically. Um, and so, there's a lot of things that go into this. Y'all will work on the pixel to speckle size, which I'll talk a little bit about that, but not go into too much detail because that'll be one part of the lab. Typically people assume this to be 1 because it's convenient. And then, but we all know what assumptions can do. So it's something that if you do it properly, you could either measure it or you should fit for it. Okay. So now we went from here, the covariance, we can get G2. From G2, we can get G1. And if you look at G1, it's the dynamics. So you're going to see these exponential decays and like in this. So it's convenient. I mean, it's convenient from a mathematical and just from a kind of, everybody knows what an exponential decay looks like. And so this exponential decay is really how the electric field will change over time as the speckles change. Okay, so if it's slower varying, so this is a correlation time here. So basically, the longer your correlation time, you could think of it as the longer the speckle stays in the state that it's in, and just slowly changing over time. Okay, so when you have that, then then G1 decays more slowly, right? It's this number, the denominator here is getting much bigger. When you have when the speckle is changing fast, this correlation time will be short, and so then this G1 will decay more quickly. Okay, so just the correlation time is effectively how long is the speckle there for. It's not quite that simple, but that's just, I think, an easy way to think about it. Okay, and it just happens to be an exponential decay. So kind of, like I said, something that we could wrap our heads around. So if you kind of put that all together, so I take this G1, I put it into this equation, for G2, and I back propagate and I put it into this covariance term, then I integrate. Okay, so I'm just going backwards now. I can get this contrast equation. Okay, so this is the canonical speckle contrast equation that is oftentimes used. T, we know, that's our exposure time. K, we measure. So the only unknown, there's two unknowns, beta and this correlation time. Beta we can measure. So the only unknown here is correlation time, which just happens to show up here 3 times. So it makes it a little bit hard to invert this equation. But then we get the correlation. Once we get the correlation time out for a given contrast measurement, we then take the reciprocal of that, and that's our inverse correlation time, or what we just term speckle flow index, SFI. By the way, LSI, most people call it LSCI, laser speckle contrast imaging. We're not doing it to be contrarian. We just use. There were all these other acronyms that went along. We don't like any of them. And LSI, I just like 3-letter acronyms. So that's just why we use it. We're not trying to be different. It's just, but we unfortunately are. Other people use it, which is kind of nice, but yeah. So LSCI is what you normally see in the literature. Okay. And this is what that graph looks like if you plot it. So if I just take this equation here, you see T over tau C in 3 places here. So a lot of times people just kind of plot it like this. Okay, so T is typically fixed. That's our exposure time. So we know that. We measure this guy over here, this contrast, and this is the only unknown. So the question I'll have is where do we want to operate in this curve? So we're measuring this over here. So where would you want to operate if you want to be sensitive to flow? I don't know. Yes. Thank you. Okay, right there. Okay. It's actually down there because it is logs, but that's okay. So if you just kind of look at these values here, so you have 0.05 and you have 2. You know, these are ballpark. These are kind of convenient right here. Okay. So you have these numbers here for this ratio right here. So we just kind of look to see where it maps to. It maps to contrast value of 0.2 to 0.86-ish around there. Okay. 0.85. So what we can do is if we have a sense of what the dynamics of our system are, we can tune the exposure time so that we try to work in this range. Okay. Yeah, John. Periodic. Why would you think it's periodic? So if you're looking at the actual, like if you're measuring directly like the electric field or the intensity, then I can see what you're saying. We're measuring we're measuring correlation at this point. So we're measuring how the intensity is changing, how it's changing over time, or how it's different, how the intensity at a point is changing over time, but at a slow is that a lower is that a lower is that a lower it's a slow is that a lower rate or lower sampling frequency than what you would have for actually sampling the I'm sorry. I think I know what you're saying, but yeah, we're looking. So it's like once they get a speckle there that's got an intensity, how's that speckle changing over time? That's what we're looking at is that how that intensity changes over time, not the actual sinusoidal pad. That's why sinusoidal wave. Okay. All right. So going with this logic, if we want it to be sensitive to faster flow speeds, do we want to increase or decrease the exposure time? Increase. Why? Oh, sorry. Decrease. Why? Okay. So yeah, so the correlation time would be shorter because we have faster flow speeds. So we want to decrease the exposure time to try to work in this range. And I think intuitively you could think about it like something's moving really fast, you want a short shutter to be able to see it properly. So it's a similar concept there. And then if we increase the exposure time by a lot, what would happen to the contrast? if you increase the exposure time by a lot, let's say you went from 10 milliseconds to 10 seconds, what would happen to the contrast? Starts to, yeah, starts to disappear. So basically, yeah, you're working from this area and you're starting to go in this direction right here. And it makes sense even if I'm kind of moving like that speed, if you have a long enough shutter time, I still will look blurry. The contrast will still go down. Okay. And then if we decrease exposure time by a lot, then this would start shooting up over here. And you can just see mathematically, this number would go down, but also I think just from a intuitive perspective. So if I had an experiment, I measure a contrast of 0.4, my exposure time is 5 milliseconds, then my correlation time is 0.55 milliseconds. So right away, I get it out. And I could take 1 over that. That gives me some speckle flow index, some flow metric. If I go over here and I have a 10 millisecond exposure and I measure 0.72, then I have a correlation time of 10 milliseconds. So I can measure flow this way. It's actually quite straight. It's quite the concept, at least from an application perspective, is quite simple. And remarkably enough, if you just take measurements from a sample and you kind of over time, like, you're measuring something over time, the relative changes actually are they do a pretty good job of actually mimicking the relative changes if you look at the actual measurement. It's actually, so this works quite well for that. Yeah, Anna. Yes. Yes. So the and this is where, going back here, data is the main thing that it would typically you would consider as affecting it. Like if you don't use polarized cross polarized light, if you don't, so yeah, so obviously noise too. Noise will change your intensity. So if you have a lot of, let's just go the extreme example, you have like just this massive offset in your information. So now instead of your intensities going like that, the riding on top of this wave, that's going to also artificially decrease your contrast. So definitely the answer is yes. It's not like, yeah, it's not one size fits all. Um, that's a great question. But yeah, a lot of this, you can kind of couch some of this in this beta term here. So room lights on, room lights off, that can have an impact. Um, okay, and this is the main equation, once again, just to show it here. And Christian talked about the simplified equation. What we showed, this is 16 years ago now. If you work at a certain contrast range, you can just basically do a pretty quick fit. So instead of having to solve this equation right here, or use a lookup table or what have you, you can do this, which you could calculate ridiculously fast. And so so if you have like GPUs and things like that, you can parallelize the contrast calculation, and then you could basically, this is trivial to calculate for a computer. So you can go real time. That's what Christian was showing with actually, he didn't show the video, but we can show it probably not without the recording. Okay, any questions so far? Yeah, Anna.
Of the contrast to find the contrast, and then you would use the simplified form of the relative to find it.
No, I mean so this is the full curve right here, right? So, and that this is going to be, this is basically it's a quadratic, so it's going to have it'll be a different shape to it, but for a large number of, like, basically this contrast, I guess we can show here. Basically, this contrast down. If your contrast is in that range, you can just use this equation. Oh, sorry.
Used to find contrast.
That's the standard deviation over the mean. So that's sliding window. Yeah. Sorry. Yeah. So like what Christian showed earlier. Okay, so this is going to lead into a lab. I'm not going to explain. I'm going to leave this part with a question, and then that's for the lab. So how large is a speckle? So, I don't want to keep going back, but there's that fully evolved pattern that hopefully maybe you remember. So it's just diffraction limited. It's the same thing as basically when you're doing, like microscopy and trying to figure out what the diffraction limited spot size is. The microscope, you have numerical aperture, you have F number, things like that. So it's following a very similar type of equation here. So this is the speckle size. you have your wavelength, which, the longer the wavelength, the larger your spot will be, magnification of the system, and then the F number, or which is related to numerical aperture. So there's nothing new here in terms of just this is the same sort of equation you would have for microscopy or just imaging in general for what's the smallest spot you can get to. And this is no different. So just that's something I wanted to emphasize. So, we mapped the speckle pattern onto a detector array. And what's the best way to sample this pattern? That'll be the part of the lab. Okay. I like to let y'all kind of discover this sort of thing, so I don't want to say anything more. Yeah. It's so the simplest way would be if you had just a single lens, it's the focal length of the lens divided by the the open aperture of the lens. So if you just had a single lens, it'd be focal length over diameter. Compound lenses get more complicated when you have multiple lens elements. I'm not about to pretend I understand the ray tracing like well enough to be able to tell you how it works, but most of those compound lenses or macro lenses will have F number as on the basically on the lens itself, and you can kind of control it. Yeah. So this is related was I think it's the Abbey diffraction limit. So it's basically with the with the it's with the Bessel function. Yeah.
How do you, so is M set by the magnification of the image system, and what is the average magnification?
That's going to depend. Most of the time we're minifying. Most of the time we're actually measure our M is less than one because we're measuring areas larger than the detector. Um, if you go with a kind of a really crude sensor size about a centimeter, they can be much smaller. But then, 1-to-1 would be a centimeter to a centimeter. So it's yeah. I mean, you could do it. You could calculate it theoretically, but it's very easy to measure empirically.
So the distance, if this is a bigger area size, then bigger magnification, smaller magnification.
Smaller magnification, yes. Yeah, so for a lot of things we do, this sum in the parentheses there will be 2 or less. A lot of times it's getting close to 1 because we're imaging a pretty large area. Okay, so that'll be up to you guys to kind of play around with. All right. So contrast, we talked about, going off with Christian showed with the sliding window. So that's this approach right here. You can also do it temporally. So what do I mean by that? But the equation's the same. It's standard deviation over mean. But what do I mean by that? So spatially would be this is just, showing a 3x3. You probably want more, but this is basically having a 3x3 window that you slide. That's your spatial. Temporally, when you take 1 pixel, you need multiple images. You take 1 pixel, and then you look at how that intensity varies over time of that 1 pixel. Okay. 15, 25 images, they're not uncommon. And you're basically looking at every point XY, you're looking at how this intensity changes over time. Okay. So in this case, like with a 5x5 window, you would have 25 points that you're sampling, 25 pixels. Obviously in this specific case with 15 images, you would have 15 points. This is just talk about an old looking image. So this is a figure from that 2003 paper by Haiyan Chang. So just laser and imaging system. You don't worry so much about the optics here or anything. So in this specific case, they took a porcelain plate and they just kind of moved it with a separate motor. So it's not biological tissue. It's pretty much a surface reflector. Okay. And this thing's moving at some known speed. Okay, so x-axis is speed, y-axis is contrast. What do you think would happen if I move along the x-axis? Contrast will do up, down, left, right, decrease. Excellent. And what's kind of cool about this is that if you look at this spatially or you look at temporally, you actually get the same contrast value. Okay. So they overlap, which is kind of interesting. I'll get into the y in a second. Oh, sorry, this is a different paper, but it's the same group. Okay, what would you expect to see if the plate wasn't moving? Fixed meaning, so spatial and temporal contrast. What would you expect to see? Straight line. Well, the temporal.
Yeah.
Okay, so what will the contrast be if it's a straight line? Standard deviation over mean. So what's your standard deviation if it's a straight line?
No, 0.
Okay, 0. Okay. Okay. Well, spatially. So I have 25 pixels right here, or 5 by 5 pixel. What would you expect to see? Would you expect it to be 0? If you have a speckle pattern on there? No. So that's what we're seeing here. So we have this slide, this is look at one point, and then we have this pixel over here. Okay. Standard deviation over mean for spatial, standard deviation over mean for temporal. So in the window, you're gonna going to what Lokesh was saying, you look at one pixel, if the thing's just sitting there not moving, and let's just say there's no noise, you can get the same intensity value at all these points. It's not changing. So this would be 0, as Lokesh said. In this case, we have a speckle pattern. So we have intensities that if it's a nice pattern, they don't vary. So there's a discrepancy here, and this is basically what you see. So for the spatial analysis, you see this value up here, and then for the temporal, you see this value down here. So basically close to 0, which would be what happened if you had no variation. So it works, and then it doesn't work all of a sudden. And so why, we'll get into the why is that, but if I just plot the spatial and the temporal values against each other for each one, I just pair each one of these points, get this nice linear fit here, and then it just drops off right here, where that's this point plotted against this point. Okay? So the theory works, and then at some point, it doesn't seem to work anymore. So one thing that you will be doing in the lab, not specific to this, but I'll get to this point in a little bit, is why would you even do temporal? Why would you even do spatial? So we're going to go into, so that's going to be one of the labs. So you'll be working with some data, and qualitatively you'll be able to look at it, and then and hopefully you can reason out a why they look different. So in this case, we're talking about spatial contrast and temporal. Spatial contrast is what we were talking about before. Now we added temporal contrast as another way to get contrast. Obviously, for temporal, you need multiple images. Spatial, you can do it with one, or you can do it with multiple. We have the same G1 term here, and then we have this contrast equation, contrast versus correlation time, which is still the same. Okay, so this is all we've done so far. This is all we've touched. So the only thing that's been different here is contrast. The way we calculate it is different, but all this stuff is still the same right here. Okay, so I think this is going well, but this is obviously it's beauty's in the eye of the beholder. So questions? Yeah.
That's not why is there a speckle pattern still for spatial? Though there's a standard deviation, there's no standard deviation.
No, for temporal, there's no standard deviation, right? So if I'm taking image after image after image, like the stack in this direction in time, and I look at one point and nothing's moving, that one point's going to be the same. But in each image, there's a speckle pattern. And the speckle pattern, like if I look at it in each image, it looks, theoretically looks identical in each image. But if I look at a region in that image, there's a pattern. Yeah, Bjorn. Mhm. No, that you could do that over the, that's called global contrast. So these are typically in small windows. Yeah, so there's a reason why you use certain sizes. And that's gonna be one of the labs actually. I keep feel like I'm just punting everything in the lab. But that, like I said, I like for y'all have some discovery with this as well. So yeah, Mitch. Yes. So people do that as well. So that's actually this diagram right here. This is actually, I think, what, so I kind of I bastardized what the message was here. Like this one was actually meant to be. You can see over there those 3x3 windows that go time. So you could do 5x5 or 7x7 in space for a single image. So that's 25 or 49 samples or pixels. You could do 1 by however many in time, or you could do 3x3 by 4 or 5, right? So there are multiple ways you can try to sample the speckle pattern. Whether you add multiple frames, or whether you do single frame, whether you do single pixel, multiple frames. So yes, that's called spatial temporal, for lack of a better term. Yeah, John. How do you deal with the 0 point? We're not looking at velocity. We're looking at speed. We like with this sort of imaging, you cannot, unless you go at really high speeds and track speckle actually moving, which you can do. They do that for biomechanics work. In general, you don't have any directionality to this. It's just basically a flow, a metric of flow, but it's not telling you which direction it's going in.
It's a scaling.
Yes, yes. Like I said, there are tricks you can borrow from microscopy to try to get the vector. And people have tried doing also like arterial venual sort of, which tend to be counterflow, with different tricks. Yeah, gosh,
What are the applications where it can
that's for the lab. I think you'll see it pretty quickly. But yeah, I mean, definitely with temporal, one of the disadvantages is you have to have multiple images, right? So your temporal resolution by definition should decrease. But there are advantages. Okay. So this isn't too bad. This sort of thing. And so this is where you start getting a little bit more, and it starts going a little bit more off the rails, but these are things that are actually we would encounter when we do speckle on most tissues. So right now, with what the kind of the equations and the underlying physics that we've been talking about, it assumes if a camera looking at some object where everything is moving, and everything's moving at one rate. So it's a homogeneous mixture of particles. You have these equations, they're largely Brownian motion based. So just things above absolute zero, you get molecular motion. And so things are just moving. So for biological tissues, when you're talking about perfusion and you're talking about capillary level flow, and we're not resolving capillaries. Like if I look at my skin, which looks kind of red right now, sunburn. Basically, I can't see the individual capillaries there for sure, but I know there's blood flow there. So and with capillary flow and with especially certain organs, it's going in all directions. So a very convenient way to think about trying to quantify flow is actually as a diffusive process. So it's not really like that. The red blood cells are obviously moving in all these tubes inside your organs, but thinking about it as it's directionless, randomless motion is pretty common with diffuse optical techniques. Like diffuse correlation spectroscopy is one of them. DOSI, like a lot of these technologies where you're trying to just get your spatial scales are way bigger than the actual spatial scales of the particles are moving in. Okay. So but this is reality of most things, right? Most things you don't have blood vessels right at the surface. The brain's an exception. Some other organs you have major vessels on the surface. But in reality, a lot of the perfusion subsurface. And so, if we just assume there's that in that red block, there's a homogeneous mixture of particles, and it's mainly Brownian motion, so diffusive motion. So the question is, if I measured this, and then I just could magically sync if I could sync this area here down, would I get the same correlation time? Would I get the same flow dynamics? And Anna's shaking her head no. So this is where you start getting into, and it's kind of an exercise in the obvious, but so Christian kind of showed this example here where you have like the different waves and everything like that. We're assuming that mainly it's all these changes here, kind of in the shifting, are due to they're due to particle motion. And in reality, you have moving scatterers, but you also have what are called static scatterers. So you have some particles that are moving at a rate, and you have some that are moving at a lower rate. So basically, two types of Brownian motion. You have something that's of interest, and you have something that's way lower. That's just, let's say, the matrix, the collagen, or the surrounding the non-vascular areas that also have some particle motion to them, just because of Brownian motion. So there's this discrepancy here. Static is kind of it's a loaded term, but we have particles that we would call static, and we ones that are called dynamic. And with blood, we'll call that dynamic. Okay. So if we go back to this, we have a kind of a model that looks like this now, where if I just assume that the light doesn't go below this red block here, everything here is dynamic. And here I have, clearly to get light here in the back, I need to go through the static layer here as well. Okay. So there's this term called ergodicity. So if we're calling speckle random, a random process, then what should happen is that the time average should equal the spatial average of the process. Okay, and we're going to assume that with a speckle. So if we're looking at a dynamic object right here, everything's moving here. Or if we're saying the pattern is dynamic and random. So in this case, and we saw with that measurement, the temporal and the spatial statistics, the contrasts were pretty much the same. So this is a case in which ergodicity is satisfied. Basically, the temporal processes and the spatial processes, they're equal. And that's what we saw here. So then when we went to the stationary one, temporal equals spatial. So in this case, so if we actually go to the full extreme, just go to the stationary object, the spatial pattern is not varying in time, right? We have a pattern on that comes back to here, but the pattern's staying the same with each frame that we got. So in this case, the temporal and the spatial statistics don't agree. And so this is the case where ergodicity is not satisfied. And so if this is an ergodic process when it's moving, but when it's not moving, it's not ergodic. And so because of this, we need to actually take into account the fact that some scatterers are moving and they follow this ergodic process, and some are not. Or the spatial temporal statistics will equal each other, and sometimes they won't. And if we think about it, if we have blood, or if we're imaging a tissue, we have these scatterers that are moving, and then we have these ones that are moving a heck of a lot slower. The neurons, what have you. We basically can model this as a dynamic and a static contribution to the electric field. Okay, so this is what we've been looking at. Now we have to add this other component. So this is actually from Ashwin Parthasarathi, who was a PhD student with Andy Dunn, who's pioneered a lot of the speckle rebirth on neuroimaging. He's got questionable taste in graduate students, so Mitch is one of his graduate students. So I say that in jest. So this is basically, so you have this time varying electric field, you have the static field, and then it's this term here just represents that as a sinusoidal wave. And so yeah, with this dynamic field, with the static field, and then this is basically relate to the wavelength of the light. Okay, so the electric field is varying in time. And so this is what we had before this is a fully dynamic sample. Basically, we had this nice exponential decay. We still have this for the dynamic regions of the tissue. The static regions of the tissue, we don't have it anymore. This is basically a constant. And so when we consider both static and dynamic scatterers, we basically got, this is the original electric field autocorrelation that so this is the one we're happy with, the single exponential, or this exponential, this term correlation time increases, this decrease, this decays more slowly, etc. But now we have to add the static scatterers. So basically we have typically a term that's used as rho. So we have if you have some dynamic scatterers, some static scatterers, then this, if you have a lot, like 75% dynamic scatters, and this value will be 0.75. And then we have the intensity that we associate just with the static, basically what's scattered from static structures. Now, we've had a long discussion on what's the static structure, but that's a topic for another time. So this was our original relationship between the intensity autocorrelation and our electric field autocorrelation. Okay. So this is with assuming everything's dynamic. But once we add this concept of non-ergodicity, and these static scatterers, this is what our equation turns into. More complex. There's some more unknowns in here as well. So we already introduced this rho. We don't know what this rho is off the top of our, or what this rho is. There's a noise term, which is just an offset. But this is basically what our intensity autocorrelation function looks like now. So like I said, a little more complicated. If we just kind of go to a limit and we assume everything's moving, so we go back to our dynamic scattering, like with that red block up at the top, this just collapses back down to the equation we had before. So it at least shows that at the limit of where everything's moving at some rate, this works. And if we go to 0, so there's no dynamic scatters. Everything's not moving. We just kind of plug that in, just just start crossing out terms. This term crosses out, this term crosses out. You basically get a constant. So meaning if nothing's moving, and you look at a point, it's staying the same over time. So it all kind of matches intuition. And then if we assume that this is the dynamics once again of this defines the dynamics of the dynamic scatterers. This is what, if you plug that G2 into the original equation, this is what you get for contrast. So it's more involved. But same thing. We know our exposure time, which is baked into this. There's this X term, which is T over tau C. So we know our exposure time. We measure contrast. Problem is there's 4 unknowns here now. Before there was just 1. Okay, but so we're measuring this and we know this. So going back to this graph right here, with the 4 unknowns, you need at least 4 unique equations. So this is what's called multi-exposure speckle imaging. So Mitch, to your point, this is basically what we're talking about here. You just go at multiple exposures on your camera. So there are different ways to do it. The way that Ashwin did it originally was they literally would change the exposure time of the camera setting, take an image, take some images, change the exposure time of the cameras, take images, etc. Now people will do things like you'll have 100 milliseconds and you need 1 millisecond, then you just take 101 millisecond images and then just basically integrate them into a 100 millisecond image. If you can acquire the images fast enough. So that's what a lot of people do now. You can't just take 101 millisecond images with big delays between and fuse them. That won't work. But yeah, that's basically what this is. So we run this experiment here where we just pump a fluid through a tube and then we're just imaging it. So this is what the contrast might look like if we just change exposure time. All we're doing is changing exposure time, mapping the images, looking at contrast of the tube. You can fit this equation to this, and you can just start measuring this for lots of different flow speeds. So this is exposure time, changing exposure time for one flow speed. This is then when you add in all the other flow speeds. Okay. So question I have, which one's 0? Would 0 milliseconds have millimeters per second have higher contrast or lower contrast than the other ones? Higher contrast. So this one's 0. And then basically it's going into at these different speeds. So this is basically, it's related variance, but basically you get this trend where it starts collapsing in on itself like this. Okay, when you increase the flow speed, you get all these exposure times. And then you can start fitting for all those parameters. And so if you account then for the static scatterers or the fraction of the fraction of dynamic scatterers, and you don't just assume it's 1 all the time, you can then get these measurements that, basically different flow speeds, and this is your SFI. Get a nice linear trend here. The thing that's kind of cool is that if you do this with no top layer, you can get an image that looks like this, or a graph that looks like this. you add a top layer where now there's static scattering and you account for it, because all our dynamic scatterers now, there's 15% of them that are not dynamic, but you account for it, you can get basically a similar trend. So in this case, you can adjust the measurement just by taking multiple exposure times, fit for more parameters, and then you can basically negate the effect of having the static scattering to then measure your dynamics. So this is a kind of neat trick that Andy Dunn's group and Ashwin that they developed. If you do this if you just do this at one exposure and then you do it here with another exposure, you're going to get a different value of your correlation time. It's just because you have the static scatterers on top and you're not accounting for them. Yeah, Bjorn. That's a great question. We've struggled with what the concept of a static scatterer is. Christian, do you have an answer to that question? So it's like, yeah, so imagine you have like 20 photons going in that and so this we've been trying to figure out how to simulate this. So you do a Monte Carlo model, let's just say you're doing it at particle level, and then it scatters 85 times. And it's by hitting static scatterers. And then one time it hits this dynamic scatter and then it goes out. Is that dynamic? Is that a dynamic photon or a static photon? Versus like if it goes in and interacts 30 times with dynamic scatterers, moving scatterers only, then you know that's a dynamically scatter, or it's a photon that's interacted with dynamic scatterers. So some is what percentage. So we've struggled with this, but that's, I guess if you really thought of it as like pool balls going through or a pinball going through a bunch of bumpers, and if it was always deterministic every time, like how it bumps off of them and it always comes out this way, and they just had one, and there were 30 bumpers here as it's going around. I just had one bumper that would sometimes be there, sometimes would not, sometimes would be there. Then yeah, it's going to change the trajectory. You see that one event. So my guess is it's a weighting. Like if it interacts most of the time with static scatters and then just once with dynamic, versus if that's going to have it's going to have an impact with less of an impact. That's my guess. It's a good question though. Okay. So this take home message then is we need a bunch of images. We take these at different exposure times. Now we get spatial contrast at multiple exposure times. That's the difference here now. Is G1, G2 equations. The G1 equation is the same. We're assuming that the dynamics for the electric field in a dynamic region are the same, but now we're adding the static component in. And so that makes the equation a little more complicated when you add this part in. And then hence it makes the contrast equation a little more complicated, but it's all been worked out. So the key is you need to measure at multiple exposure times, calculate the contrast at each of these times, and then do fitting, in order to try to get the other parameters out. Okay. So this is multi-exposure speckle imaging. All right. Quick question. Why the photon go to therapy? Trying to wake people up here. Oh, he was losing a sense of direction. All right. So going with sense of direction here. Any questions? Last year I had dad jokes every morning, and I think they got a little tiring after a while, but I'm a dad, so it's okay. Christian, have you gotten into dad jokes yet? Not yet. Okay. Yeah, Lukash.
When there are no dynamic scattering, G2 is 0, right? The G2 is constant.
Sorry, when there's no dynamics. Oh.
Yeah, G2 is only.
Yeah, your correlation time should be infinite. So.
Yes. So what happens to
G? G1 will also be, it'd be because your electric field is static at that point, so it's just constant. Yeah. So if you really have conditions like that, the G1 and G2 will decay based on noise only. Yeah. So. If you were to run an experiment with this technique here, then you would need to, if you wanted to, then you would need the multiple exposure times. Now, there are some papers from Andy Dunn's group on what are the optimal selections. Like you can kind of be brute force and just pick 20 exposure times, but there's also some logic to which ones you might want to pick more. There's obviously more sensitivity to certain exposure times than to others. But yeah, that's what you would need to do if you wanted to use this technique, is you have to get at least 4 different exposure times. And then how you get the different exposure times, once again, that depends on, they use an acousto-optic modulator. And so, oh yeah, I said the wrong thing actually before. They didn't use different exposure times on the camera. They set the exposure time to a long value. And then they use an acousto-optic modulator to deflect the beam for certain periods of time. So, and then they use, I forget, I'm trying to think I remember how they control the intensity. That's what they own too. Okay. So that's how so they did it on the excitation side, not on the imaging side. Yeah. Cool. You have to, so they were trying to keep the overall intensity equal. So they would deliver at the shorter quote unquote exposure time, they'll deliver more light. So the integrated intensity was the same for each of the exposures because they didn't want that to be a factor as well. So you have 100 millisecond exposure time, but then only for 100 microseconds is it actually exposed at a relatively intense amount of light versus that 10 milliseconds you expose for 10 milliseconds, but with less light. So that's how they controlled it. Yeah, if you did it without adjusting for that, you would have to figure out your longest exposure time, what's not saturated, and then back off from that. But the problem there is that the short exposure times you don't have much light. Now one of the jokes we have in the lab is we call it the Creuset limit. So Christian's been like with this system in particular, he was working with like the very feeble number of counts, like 10 counts with 250 on a 8-bit camera, and it still worked remarkably well. Like Speckle it is an extremely forgiving technology, especially once again, all you care about is relative dynamic, and you don't necessarily care to be this quantitative. It's actually pretty forgiving. Okay. All right, let's see here. Okay, one of the things, I guess, optical properties. So Christian alluded to, I think multiple times I was going to talk about this, I'll talk about it. So if I have a solution here, and let's say I know the dynamics, and then I just snap my fingers and the absorption coefficient increases by a factor of 2. Or I snap my fingers and scattering increases by a factor of 2. What happens? Do I get the same correlation time? Ideally, yes. Then we don't have to worry about this. So the fact I'm talking about it means probably not. So this is an experiment. So David mentioned Aman Mazar, who is a PhD student here. So Aman worked with us on this specific project. I think it was when he was doing a short postdoc with us. So we had 2 containers with intralipid, and so it's a pretty simple experiment. Just change the amount of scattering in one of them. So this is the range of reduced scattering coefficients, or change the absorption. Okay, I don't think this was nigrosin. This I think was just dilution based. I don't remember off the top of my head. So in an ideal world, if I change the scattering, contrast wouldn't change. So this is actually this is actually supposed to be, sorry, this isn't supposed to be tall. This is exposure time on the x-axis here. So that increased exposure. So I'm basically writing down that speckle contrast versus tau C over T curve. I should get to lower contrast. So this should go down. Same thing over here. Okay, so that's what should happen. This is what happens. So if you change the scattering, you start getting a change in contrast. You change the absorption, you start getting a change in contrast. So why? When you change, so there's a liquid phantom, you change the scattering. You start increasing the scattering, your contrast goes down. Why do you think that is? So you're increasing scattering. So by definition, you should have more scatters in your solution. So why do you think it's going down? I'm sorry. It's more blurry, but it's more blurry because more scatterers. Okay. So yeah, so you basically have increase your scattering, you just increase the number of interactions. So it'd be like in this room, if all of a sudden, if I'm just walking through this room, just I could care less if I'm bumping into you or not. But then if I add 3 times the number of people on here, I'm going to bump into a lot more people, right? And so so I have basically more collisions at that point with dynamic scatterers. This is assuming that the particles are moving at the same speed. Obviously, if you go make this too thick, probably be moving at slower speeds. So this is a somewhat dilute solution here. Okay. I increase absorption and I decrease my contrast. Why? Yeah, Bjorn. You're decreasing the penetration depth. And so and you interact with fewer scatterers. Exactly. So this would be like if I walk once again, if I just somebody would blindfold me, I just start walking through you all. I'm going to hit a whole bunch of you, maybe some intentionally. But if I only go for a shorter depth because I can't go that far, I'm going to I should by definition then hit fewer of you. I'm getting in trouble for this, aren't I? Someone's gonna take this and just put it. I hit people. Okay. Um, you get the point. All right. Yeah. So that's exactly it. So this is gonna be part of the lab too. So I'm not gonna talk anything more about this either. Um, I will talk about how we can deal. So you're gonna kind of just work with. We have some phantom. We had some speckle images of phantoms with different optical properties. Okay. So how do we deal with this for speckle? So in this case, we borrow basically some concepts from dynamic light scattering. So some of this is borrowed from diffuse correlation spectroscopy in particular, and diffusing wave spectroscopy. So as Carol and as Vasan and as multiple people have talked about, diffusion approximation, you use it to model radiance transport inside tissues or in objects. We're going to use, and this is based off of work that David Boaz and Arjun Yo did a long time ago, but it's still quite there's a lot of utility to this. Is we're gonna look at correlation diffusion. So we're gonna look at momentum transport instead. So momentum transport is something that's follows similar sorts of equations to heat to mass, and so we're gonna use that as our state that we're basically using as for transport. And so in this case, we're actually using instead of a radiance term here and here, we're using the electric field correlation function. So we're having correlation transport. And so we're going to then, and we have this mu effective term, which is an attenuation coefficient. So I think that was Basan's lecture where he was talking about this. So it's got got absorption, it's got scattering inside it. So the kind of concept here is that you have light in this case of you have this marble going, it hits this marble right here. This is going to go in a different direction. So we already have a change in momentum here, and then we impart momentum over to this particle here. So you do this a lot of times, you can get momentum, you can get transfer momentum. You can model it as a diffusion process. And the angle is going to matter. So each scattering event will lead to momentum transfer. And each momentum transfer event that occurs with a moving particle will lead to a reduction in correlation. And this G1 term, this exponential decay, basically it reflects this loss of correlation. We've already seen that. That's what the electric field, basically the correlation time associated with it. That's what we're looking at. So this is work that Dave helped us. Dave Boaz helped us with quite a bit. And this is based off his dissertation work. So absorption coefficient you can model is a loss term. You lose light that way. You can also integrate loss due to motion, basically correlation loss with this term right here, which basically you have the wave number, so we can calculate that. We also have it based off of how for a diffusion process, root mean square displacement that's occurring. And so for Brownian motion, this is the term this is just for any particle, would be basically the 6 times diffusion coefficient times some time after the moment that the initial moment you're considering. Okay, so basically what I'm trying to say here is that we have absorption, and now we have a motion term here that's also we're calling it all this lump sum of a dynamic absorption coefficient. It's a loss term. So the reason why we do this is because if we can get this diffusion coefficient out, we can then, if I measure something on different days with a normal speckle system, and let's just say the system moved, a grad student or an undergrad bumped the system, and you have to just like with any other the microscope or anything else, it can get out things can get out of alignment, and things aren't going to perform the same way as they would have the previous day, and so because of that, just because of the behavior of the system and stuff, this thermal noise, things like that, there could be differences, and so if you just look at relative changes, that can then be misleading. So if we had something that's more on a quantitative scale, more absolute, we can then get diffusion, in this case, a diffusion coefficient. We theoretically can then measure things among different systems, different subjects, dynamic tissues, like if there's wound healing going on. Now, I put independent with a star next to it because we're never can be independent of the tissue conditions completely. This is just our way of trying to drive towards that sort of approach. At least theoretically you can, in practice, probably not. But hopefully gets us one step closer. So the electric field correlation function now takes on a very different appearance there. It's not just the exponential decay. So now we have, in this case, we have all the optical properties baked in. We have the dynamic absorption coefficient. So we have this term right here baked into the effective attenuation coefficient. We have also spatial frequency. So in this specific derivation of this was trying to do spatial frequency domain imaging along with speckle, and actually in one device. You could also just get the optical properties out, and you could start trying to and assume that your illumination is planar. So you have zero frequency, so this term cancels, and you can do this analysis in the same way. So you can try to do it with structured coherent light, which is what I show here. This is actually with a laser that then gets expanded, hits this beam splitter, goes off of this display here, which is a spatial light. It's a functioning as a spatial light modulator. So this you can basically program in different phase delays into this device, and you can then get structured illumination coming out onto the tissue surface. So we're using a laser as our light source, not a lamp, as you would with spatial frequency domain imaging. So you can do this directly. What we currently do, this is tin system, and I think, Christian, are we bringing this tin system to the? Oh, we're not. Okay, we can maybe show it another time. So this is a system that we have two of these systems that basically have the speckle arm and then the spatial frequency domain imaging arm. In Christian's case, what he's been doing is not using this. He's been using, well, for that small sensor that he showed, he was basically showing the spatially resolved diffuse reflectance spectroscopy. So any way you can measure optical properties at the wavelength of interest, combine it with the speckle measurements of contrast, you can then try to get towards diffusion coefficient. So in this case, you could do reflections at different spatial frequencies. So this is actually an example of laser illumination through this spatial light modulator. So we have structured coherent light. You could also do it separate measurements, what have you. So you can basically get these speckle contrast maps, and then you can get also you basically do the fitting to this electric field correlation function. And basically we do numerical integration typically with this equation in a form like this to then get the contrast out and then get the flow. We measure the contrast. The diffusion coefficient is baked into this part right here. So we fit basically that's the only unknown that we're trying to fit for. Okay. Why did laser speckle pattern fail its presentation? Lost coherent can present a clear picture. So that in mind, questions. Sorry, I realize this is getting a little bit more esoteric. Trying to be more quantitative, but that's the penalty. Conceptually it gets more challenging. So kind of recap, you can do single exposure and just measure contrast, use that really simplified speckle imaging equation, bam. You get a correlation time, you get a flow map. You can do it spatially, you can do it temporally. You can then, if you want to be more quantitative with it and account for static scattering, you can then do multiple exposure times and then fit and try to get out the correlation time that way. That works quantitatively better. Or you can do something where you're trying to measure the optical properties and correct the speckle contrast measurement for changes in optical properties or differences in optical properties. Yes, I don't know. Well, Christian showed that, right? Like, I think if you have that one example is the red and blue curves, where basically like with the pressure cuff. So, for big excursions and absorption, it can have an effect. If the absorption changes are not that big, then it becomes a little bit less important. I think if you're trying to look at tissues that have wildly varying scattering properties, either that they're changing over time, or just you're looking at a map and you have a tumor here, and then I think there you might need to consider it as well. But if I'm just looking at normal skin over a period of days, and I don't think scattering is going to change that much, it's assumption. I mean, like I said, I'm sunburned, so my absorption's higher now. This might actually play a role, but if I'm just looking day to day, probably doesn't matter. Thank you. So that's really what it comes down to is that you have to first really define what you want to measure, how well you want to measure it, and then you pick the speckle tool. If you're going to use speckle, you pick the speckle embodiment that you need. So yeah. Okay, this will be, I think, the last thing I talk about. Can we be more assumption free? So going back to our friend here, we're going to return back to our friend here. This nice simple G1 term. So what's better than one correlation function? So in this case, you tweak it a little bit. So to actually break apart the Brownian motion into two different correlation functions. So already the answer is partially here. 3 correlation functions. So you have the middle one is the one that we've been using. This exponential. But you notice they're all exponential decays. Just one's a square root, one's power of 1, and one's a power of 2. That's it. It's actually pretty amazing that the behavior can be modeled that relatively simply. So the Brownian motion term is, why am I pointing at the screen there? Brownian motion term basically gets split apart into either you have mainly Brownian motion. So there's a little bit of a tweak here in terms of what sort of dynamics this one's actually mapping to. So you can basically start separating it out, whether you have some healthy mix of ordered and Brownian motion, you have mainly Brownian motion, or you have mainly ordered motion. So ordered motion would be large vessels, for instance. So this is a Gaussian type of distribution here, and this is a Lorentzian, and I forgot what this one's called. So this is now basically doing imaging. This is trying to do imaging more like DCS, like diffuse correlation spectroscopy, where you have, basically you do laser illumination at one point, and then take measurements really fast. You're trying to you're trying to image or measure the correlation function directly. You're trying to measure G2 directly. In this case, we're trying to do the same thing. We're just trying to do it now in an imaging modality. So we have, I don't know, however many pixels, let's say 3 million pixels, doing 3 million basically DCS measurements. We don't actually, you can't go that fast with the camera we have. But unless you window down. So I think I forgot how many, I think we get like a few thousand. Like maybe like 20,000 pixels. So that's all it is. You just have a high-speed camera. So we're going fast now. So you're actually measuring this intensity fluctuation fast. Okay. Then like I said, and then you have intensity, and you're basically calculating G2 directly. So no assumptions being made on what G2 looks like right now. You basically just calculate, you're measuring, and then you're basically and then you're calculating G2 directly. So these are the sorts of, this is a mouse brain. So this is work from Dmitry Postnov and Dave Boaz. So each one of these points is basically you're getting the G2 function out directly. So this is looking at what the image looks like at 0 microseconds. This is what looks like at 440 microseconds later. So it's changing over time. And changing at different rates depending on the flow dynamics. So it's going to change So once again, G starts here and then goes down. And it's the rate at which it goes down is going to represent how fast things are changing. So you can see like in this large vessel in the middle is already a lot lower in terms of the G2 value than it is over on the side. This is kind of showing if you look at different points of time. So the two vessels, like the two large vessels are down here, so they correlate faster. I think this is modeling, but this kind of shows it. And then this is looking at an area that doesn't have an obvious vessel with it, and it's decaying slower. Okay, so you're measuring this directly. And this is our fun G2 equation right here. All this is an extension of the multi-exposure equation, but now it's just got terms related to different types of flow dynamics. So basically, it's taking these three correlation functions and integrating them in as fractions. So I have 25% going at Brownian motion, or at Lorentz, basically at G1 at an exponential decay, and I have 45% going at an exponential decay squared, and then I have the remaining 35% going at the other distribution. That's all you're trying to do is basically try to determine based on fitting which dynamics dominate or which dynamics you have, what the fraction of them are. Okay? Yeah, Mitch. I'll get to that in the next slide. So there's a 0.5, a 1, and a 2. And what those represent to are the power here. So this is to the 0.5, this is to the 1, and this is to the 2. So it's basically what each of those components. So D is basically the relative fraction of one component. And, 1 minus D would be another one. So this specific form of it, you're basically mapping to just two correlation functions. And the same thing, you have your fraction of dynamic scattering, that's still here, rho. 1 minus rho is obviously fraction of static scattering. You have a noise term, you have beta. So a lot of the terms, a lot of our old friends are still here. It's just now we're adding in more correlation functions. And if we put in 1, it all collapses down. So if there's all dynamic scatterers, so going back to the simple single exposure speckle imaging thing that we started with, you basically got secret relation right out. There's also an offset, but it's all there. So this is what we ideally would love to have, but in reality, we're going to have something that's less simple than this. But you could always run with this and get some data out of it. So kind of showing the different things that, so I had to address Mitch's question. So we have the squared to the 1 power, or to the power of 1, and then power of 0.5. So those just represent different dynamics. So in this case, this is just color mapping to the different types of the different dynamics. And it's kind of it's a little bit of a confusing graph here to show, but the red is basically showcasing the large vessels. The green is some transition between the large vessels and the smaller vessels. And then this blue-black area here is where you have mainly the areas that don't have obvious vessels that you can image. So it's called the parenchyma in the brain. So this is kind of a way to segment out the different types of flow dynamics you can see based on fitting that equation. You measure the intensity of each pixel, you get the G2 value out from doing correlation on that intensity, then you fit to that G2, and then you can back out different parameters. The fitting takes some time. So yeah, so in this case, we're just doing reflectance imaging at high speeds. We're getting a lot of images very fast, short period of time overall. We're basically assuming three different correlation decays, and we're getting out. We're measuring G2, and then we're basically then doing fitting to try to get out, at the end of the day, some sort of flow value that's corrected for all these different things going on. Okay. This one's really short. This is just showing, we talked about correlation transport or correlation diffusion. So what we can do from, yeah, Claudia. So those are the sorts of dynamics that they basically effectively map to. So when you have a correlation, so when you have the lower one in particular, that is a form of the electric field correlation function that is it is for single scattering from ordered flow. So that's the type of dynamic it represents. Before we had this as Brownian motion, but they and others basically, in 2005, this is kind of a really underappreciated paper in Spekly. But it basically tried to parse out this term for Brownian motion into a finer, basically into two different components. Okay. All right, so just the other way we can do is we can just model, we can basically be completely assumption free. We can actually measure, we can calculate the momentum transfer that's occurring with each of these collisions, right? So we know what it's a new scattering angle. We can basically tally that up in Monte Carlo. And then if we just kind of get this distribution of scattering, we get the distribution of all these different scattering angles and these momentum transfer events that are occurring, we can basically just get to this G1 equation where there's no assumption being made now. We're basically just tallying up with the model. So you can have pretty complicated structures at this point when you're modeling it. You got different layers. You can have, and we can go up to 2 different flow speed differences, but you can basically, just by running Monte Carlo, looking at the types of interactions that you have at all these different layers and with the light coming out, you can basically then, with these different momentum transfer events that are occurring, you just basically create a histogram out of it with basically P of Y, and then with some width for, you have 30 bends, 40 bends, 100 bends, what have you. You just tally up the number of times the momentum transfer goes in each one of these, and you can basically then get your G1 from that without any other assumptions being made on what it's not exponential. It's going to be some distribution that's based off of what we see with the or what we get with the from the model. So an example, and this is the last thing, this I think is literally the last thing I'll show, is so this is more from Caitlin Regan when she was working in the lab. So this is trying to mimic skin. So we have an epidermal layer, avascular layer, blood layer, avascular layer, blood layer, or low vasculature. And so if you just kind of look at this at different wavelengths, so this is some of the stuff that led to the some of the work looking at speckle versus SPG versus PPG with different skin types. So if we just look at the momentum transfer that occurs in each of these different layers. Now, if you compare 375 to 488, 488 you have a lot more blood. You have you have more momentum transfer that's basically occurring at the blood layer. The light's going deeper, first of all. So basically you can interact more with this blood layer right here. 633 goes much deeper, but if you just look at this scale is way smaller than this. So basically the momentum transfer that's occurring at those points is a lot less, actually. Wait, actually, that's not right. I think this got covered. That doesn't make any sense. Okay, ignore what I just said. But basically, as the light goes deeper, you can have more correlation. You can have more momentum transfer events. You can model it this way in a kind of a complicated structure. So we can also do it that way too. We can basically model the process and basically look at the different momentum transfer events and tally it up to then get the G1 value that way, or G1 function that way. So with standard LSI, the simplest version, you can get, in this case, we have all dynamic scatterers, and it's a fairly simple equation that we can solve to map contrast onto flow. If we have to deal with static scatterers, we do multi-exposure speckle. We need these multiple exposure times in order to be able to back out the fraction of dynamic scatterers, because it's going to be less than 1 at this point. And we can basically model the correlation function in this way. We can do LSI plus SFDI or LSI plus whatever optical property measurement you want to do. And you can get a contrast. You can basically get the G1 function that's more complicated, but you can basically numerically integrate that function in order to get map contrast to get a diffusion coefficient out. So something more quantitative. you can measure the correlation function. Or you can measure the correlation function directly and then try to match it to one of three, in this case, three different electric field correlation functions, G1s, or you can model it. So you can basically do all these different procedures, ways at depending on how complicated you want to make it and what you want to measure. Those are the kind of different ways you can look at speckle. And there's gonna be some other ones that come out, I'm sure. And there's some I haven't covered. But I think this hopefully just kind of it covers a good chunk of the space. So just I guess last thing is with the future directions. So I think the momentum transfer Monte Carlo, basically trying to model speckle as part, basically as with using a typical Monte Carlo infrastructure to it, which is what Kalen did. It needs a lot more work. We've used it a few times, but there's a lot more work that could be done there. I know Vasan has mentioned doing electric field Monte Carlo and really trying to look at it in a more rigorous manner from that perspective. Because this is, at the end of the day, it's an interference effect. It's a dynamic interference pattern. There's a lot more work on the high speed imaging, and you could do lots of different ways. There's lots of new detectors coming out. And really just kind of doing comparisons of these different techniques, I think is critical. And then I think on the more simple scale, and this kind of dovetails into what Christian was talking about, really miniaturization, multimodal, multi-scale integration, whatever you need for the clinic. Once you don't always have the luxury of time, you don't have the luxury of necessarily complex technology, complex form factors. So as simple as it requires, or as is required in order to collect data in the clinic. But you can see how simple it could be. I mean, the CSI sensor, Christian, how much does that cost now? Like $200? Yeah. So you can do this really, really cheaply. And then, as Christian alluded to, the pulsatile analysis, there's a lot that could be done there. We're really at the tip of the iceberg. So simple form factor on a finger or some region of the brain or what have you, just look at how that waveform changes over time. There's a lot to mine there for sure in terms of health. So sorry, went a little long. Hope that was okay. And I will take any additional questions.